So in order to understand how a DNA molecule works, it's important to understand its structure. Like anything in chemistry, if you want to know how a molecule works or what it does, you've got to know how it's made. So we're going to take a look at DNA, the structure of the molecule. To start with, DNA is something we call a polymer. A polymer is a long chain-like molecule. It's made of small repeating monomers, small molecules that are all attached to each other to make one big molecule. Sort of like beads on a string or train cars. And once you take the train cars and you couple them together, they become one long train. It's one big thing. So DNA is like that. There are a lot of polymers that you may be familiar with. Things like nylon or rayon or polyester. Those are all artificial or synthetic polymers. But you also have polymers in your body. Things like proteins, starch, cellulose. Cellulose is plant fibers, like cotton, for example, is cellulose. Silk, which is a protein, really, that's a polymer. And hair. Those are all natural polymers, or biopolymers, polymers of life. And DNA is just an example of that. The small molecule that repeats over and over again in DNA is called a nucleotide. The monomer of DNA is called a nucleotide. And so we're going to take a look at how a nucleotide is built. To remind you, the three components of DNA that we've studied are deoxyribose, that's a sugar. You know it's a sugar because it ends in the letters OSE. Anytime a molecule ends in OSE, it's a sugar. And deoxyribose is related to the sugar ribose, but the prefix deoxy means we've taken away an oxygen. So it's like ribose minus an oxygen. Then, of course, we have the bases, adenine, guanine, cytosine, and thymine. We'll take a look at those. They're nitrogenous bases. And finally, we have phosphates. So to take a look at the sugars first, the two sugars that you'll find in nucleic acids are ribose and deoxyribose. Ribose is found in RNA, ribonucleic acid, and deoxyribose is found in DNA, deoxyribonucleic acid. They both end in OSE, they're both sugars. Sugars are molecules made of carbon, hydrogen, and oxygen, and sugars have lots of these OH groups attached to them. Those are called hydroxyl groups. The only difference between ribose and deoxyribose occurs at the second carbon in the ring. We call that the 2' carbon. On ribose, we have attached to that a hydroxyl group. On deoxyribose, in the same position, we don't have a hydroxyl group, we have just a hydrogen. It looks like we've just taken away the oxygen, but really we've replaced the whole OH group with an H. But the net result is that deoxyribose is missing an oxygen, hence the name, deoxyribose. The carbons in the ring, 1', 2', 3', 4', and then the one that's outside of the ring, 5', are all very important in building DNA, except for the 4'. The 4' doesn't really do anything. But 1', 2', 3', and 5' are all important. And the significance of 2' is that that's where the difference is between ribose and deoxyribose. The nitrogenous bases, they're called nitrogenous. You see in the word nitrogenous the word nitrogen. They are called that because they contain lots and lots of nitrogen. And they're broken up into two families. The first are called the purines. Adenine and guanine are members of the purine family. Purine is a molecule all by itself. It's called a parent molecule because there are a lot of variations on the purine molecule structure that make new molecules, but they're still all members of the same family because they have a similar structure. Adenine and guanine are purines. Purines have 2 rings. So adenine and guanine are both double ring or bicyclic molecules. What's different about them are the things that are stuck onto the purine molecule. For example, in adenine, up here we have an NH2 group. That is called an amino group, and you've heard that word before in the word amino acid. An amino acid is called an amino acid because it contains an NH2 group, an amino group. In guanine, we don't have an amino group in the same position. It's moved a little bit. It's down here to the left. But we do have this oxygen double bond to what looks like nothing. In organic chemistry, the place where a line bends or where a line ends is usually where a carbon is. And so there's actually a carbon there. And so what we have is an oxygen double bonded to a carbon. And that group also has a name. That's called a carbonyl. So it's these additional groups that have been stuck on the purine molecule that make these two bases what they are, adenine and guanine. Similarly, the other two, thymine and cytosine, they belong to a family called the pyrimidines because they're based on the parent molecule, pyrimidine. There are a couple of functional groups that you probably recognize, and maybe there's one that you don't recognize yet. In thymine, we have two carbonyl groups. Remember, that's a carbon double bonded to an oxygen. In thymine, we also have a CH3 attached. That's called a methyl group. In cytosine, we have no methyl group, but we do have one of the carbonyls still there, and we have our friend the amino group on top. And those groups, those are called functional groups, and they give thymine and cytosine their particular properties. So adenine and guanine are the purines, thymine and cytosine are the pyrimidine bases, and they have to do with the number of rings. In order to make a nucleotide, we have to first make a nucleoside. Nucleosides are very useful molecules beyond just building DNA. The one we're going to do is we're going to use deoxyribose as the sugar, and this is over on the left here, and adenine as the base, but we could use any base. We're going to interact the hydrogen that is attached to adenine and a hydroxyl group that's attached to the 1' carbon in deoxyribose. And if you look inside the box here, we've got a hydrogen, and then we have an oxygen and another hydrogen. So that's two hydrogens and an oxygen. Well, that's water. And if we take that out as a water molecule, we call that dehydration. Dehydration means to remove water. We can create a bond between the nitrogen in adenine and the carbon, the 1' carbon in deoxyribose. And what we create is something called a nucleoside. It's now one big molecule. It's called deoxyadenosine. Deoxy from the sugar and adenosine, the aden part from adenine. That's a nucleoside. Nucleosides can be used to do a lot of different things in biochemistry. But we're going to continue to modify this to make a nucleotide so that we can build DNA out of it. In order to make a nucleotide, we can now take our deoxyadenosine and we can bring in a phosphate. A phosphate essentially is a phosphorus surrounded by 4 oxygens, and sometimes there are hydrogens there attached as well, and sometimes there aren't. It depends on the pH of the system and all that, and it's not really important. What is important is that we can do this interaction between one of the hydrogens on the phosphate and the hydroxyl group on now the 5' carbon of deoxyribose. And again, we can remove water, dehydration, and we can create a bond between that 5' carbon on deoxyribose and the oxygen of the phosphate. Now, commonly we call this adenosine, but its real name is deoxyadenosine monophosphate. Deoxy from the sugar, adenosine, the aden part from adenine. And we've just put one phosphate group on there, monophosphate. Deoxyadenosine monophosphate, or just adenosine monophosphate, is abbreviated AMP. And if I were to put 2 more phosphates on there, it would be called adenosine triphosphate, or ATP. And ATP you may have heard of, the energy molecule. But we're not going to make ATP. We're going to use this as one of the nucleotides to build DNA.